Thanks so much, Melinda, and thanks to the organizers for inviting me today as a moderator. I want to welcome our panelists for this session on GEMS in the environment and biomanufacturing. Today we're joined by Kent Smith, who's an industrial microbiologist and the current director of contracts R&D at Biomason. We're joined by Spencer Diamond, who's an associate project scientist at the University of California, Berkeley, in the laboratory of Jill Banfield. And then we are also joined by Jay Fitzgerald, who is the chief scientist for bioenergy technologies at the US Department of Energy, as well as the program manager for the BETO data modeling and analysis subprogram. So you'll hear from each of our panelists about their work in just a moment. We're going to cover a wide ranging set of applications for GEMS in the environment and manufacturing. And then we're going to talk a little bit about governance writ large, inclusive of regulation, but also of other non-regulatory aspects of governance for GEMS and in the environment and biomanufacturing. So I'm going to ask each of our panelists, starting with Spencer, to spend up to 5 minutes to give us just a bit of an overview of the areas of application that you work in relevant to our conversation today. We'd love to hear how you're applying these technologies, what stage of research and development you're in, where you're working, closed systems, environmental releases, and what types of GEMS you're using. Are these adapted wild types, gene edited organisms, GMOs? Are they derived from synthetic biology? So I'd love to just hear that quick overview of what you're working on, and then we'll get more into that kind of governance aspect of how you're doing it. So let me turn to Spencer first. And Spencer, I'll just give you a quick 1-minute warning when you're 4 minutes into a comment. Please go ahead.
All right, thank you very much. That was a really nice introduction. I'm Spencer Diamond, and as you heard, I'm a project scientist at UC Berkeley, and I'm working in Jill Banfield's lab, where primarily for a very long time, the efforts that have been undertaken here to study microbial communities in natural environments, and basically using this technology that Jill pioneered called genome-resolved metagenomics, where basically we're assembling and reconstructing the complete genomes of all of the microorganisms that we can detect using shotgun DNA sequencing in an environment, with the hope that we can understand kind of how biogeochemical cycling takes place and kind of what interactions are occurring and how nutrients are shared. Because our understanding is that microbial communities really, no individual microbe is doing what microbes do for the environment. This is kind of a collective behavior that results in kind of like a global phenotype of any given microbial community, be it in our gut or anywhere else. And so for many years, we've kind of just acted as like bird watchers, I would say. Like we're kind of just standing around and looking at the environments and kind of seeing a snapshot of what's going on. And so in the past couple of years, we've established a couple new initiatives where what we're really trying to do is affect direct genetic editing within microbial communities, specifically for the purpose of understanding functionality. So this, in theory, could have a wide range of applications in the future. But currently, what we've done is we've kind of really taken the first steps into this new area where we have some communities that we've grown in the lab. So normally we've been going out to the field and taking things like soil samples or water samples, but we've done some very, very simple synthetically constructed mixed microbial communities in the lab, or some very simple enrichments, where then we've taken the first steps into applying CRISPR-Cas technology in a collaboration with Jennifer Doudna's lab with metagenomic sequencing and some very specific types of analysis to both kind of screen which microbes might be amenable to be delivered genetic material, and then also using a specialized vector system that we developed, specifically targeting locations in the genomes of specific microbes in these communities to deliver DNA. And this has all been very proof of concept at the current time, and we're very excited about it because it's probably the first time that anyone has successfully targeted a genomic location in a mixed community of microbes and delivered DNA this way. And so this is kind of like what's happened. This is all occurring in the lab. This is very much for research purposes. This is not anything that we would be ready to deploy kind of a field setting. And I think our first question is really, can we understand microbial communities? And I'll kind of add one thing about this, in that in thinking about microbial communities, one of the things that's come up is like microbial communities are kind of like they have their own biodiversity, just like the rainforest, and we have stewardship of the rainforest. And so one thing I like to think about is, as we kind of go into this space and we're opening up this new area of trying to do genetic manipulation on communities as a whole, just the communities themselves, and even kind of the impacts that humans have on doing things like fertilization of fields or pesticides or anything like this. Are we considering, just at a very high level, what are the microbial communities that are there? What are they doing? Are we being good stewards of them? And then, thinking about that even before introducing something like that into the environment. But yeah, and so I know I said a lot of things, and my time is kind of coming to an end.
Okay, 1 minute. Any last thoughts? Yeah.
Just want to open it up to, any types of questions later, as to like both from the side of understanding microbial communities and like their function and kind of their native wild types and the stewardship of them, as well as going into now with the capability to start to affect direct genetic edits in these communities. How might that have an impact on how we decide to deploy these things and where, and also how it can answer questions about just their functionality in general.
Great. Thanks so much, Spencer. Really appreciate that introduction and that analogy to the rainforest. And let's go to Kent Smith next to introduce his work. Kent?
Yeah, thank you. I'm Kent Smith, Director of Contracts R&D at Biomason. Our company is, I guess, the world's first that use biology to make construction materials. So we use native wild-type bacteria, soil bacteria, species Sporocercina pasteuri, to build out calcium carbonate as the cementous material. So, we have industrial efforts in producing tiles of various shapes and sizes. We're moving into more larger format objects, walls and barriers, and that's all kind of confined within an industrial confined space, and those products are considered biologically inert when they leave the factory. On the other hand, in what I'm leading for the company is our exploratory government contracts work, which has been focused on a series of DARPA and Air Force contracts, really focused at field applications. So we're taking the biology of MICP and applying it at scale in the field. And to date, it's all been wild type. So we don't have to navigate the concerns of the genetic manipulations and the regulatory frameworks and environments around that. But our experience has been interesting. And when we take large volumes of our wild bacteria and deploy them in these various locales, oftentimes it seems like we're trying to learn on the fly with the regulators, with USDA, with EPA, and kind of developing the rules of the game as we go. And those will always only get more complex and more involved when taking into consideration any genetic manipulations, of course. But we also have, in addition to the ongoing active field work, we are starting to engage in more strain isolation work, more synthetic biology approaches, development of chassis strains, et cetera. And we hope that our experience within engaging in these field applications with wild type organisms will be valuable in setting precedent and helping formulate a roadmap to more complex type of engagements. So yeah, in a regulatory sense, I think one of address our experiences is just the consistency and the integration from federal level and state and local levels, because there are wildly different concerns and permitting mechanisms depending on where you are and what you're planning to deploy. So there is some definite streamlining that we see that could occur in the future to help promote what we believe has great potential, not just in what we're doing in Biomason, but biotechnology at large in the field at scale.
Great, thanks, Ken. It sounds like you had a little time. That's great. We'll save it for discussion. Let's hear an opening comment from Jay Fitzgerald.
Sure. Thanks, Julie. Really happy to be here. And thanks to you and Belinda and Anastasia for the opportunity to speak with you all today. I'm Jay Fitzgerald. I'm the Chief Scientist for the Bioenergy Technologies Office at the Department of Energy. The Bioenergy Technologies Office focuses on making fuels, chemicals, materials, and products using bio-based feedstocks and wastes with the goal of lowering greenhouse gas emissions across transportation, industry, and agriculture. Another way to kind of think about this is that we want to transform the carbon that we have into the carbon that we need for all of those different end uses while doing so sustainably. And the sources of carbon that we look at can be extremely varied, and each kind of requires a different approach. For example, we work on things like agricultural residues, forest waste, municipal solid waste, including things like plastics, purpose-grown crops, and industrial waste gases. And each of those might be transformed into the final product that we want through a different type of microbe, or even by not using microbial conversion at all and using thermochemical or catalytic processes. For today, I'm really going to focus in on our microbial conversion work, and specifically what we're doing with genetically engineered microbes. One of the greatest things about using microbes is that they've evolved over billions of years to want to consume all sorts of different types of carbon and make it into products that are useful for their reproduction. This created an incentive to funnel carbon into a limited set of biological intermediates, which can then be used to make a vast array of end products that an organism would need to survive. That can span things like energy storage, chemical defense, energy generation, and signaling compounds. As bioengineers, hopefully we can direct that microbial energy into the end products that we really want and cause them to make those preferentially over types of products that we don't want to see. So many of the projects that we fund work on trying to expand kind of the mouth of a biological funnel by combining different pathways to assimilate carbon into one organism. For example, we can engineer a strain like Pseudomonas to simultaneously consume all sorts of different types of biomass sugars or different types of lignin compounds and funnel those into a single product. So just to highlight a couple of specific examples of projects that our office funds that I think are doing really exciting things in the biological engineering space, we have a large consortium called the Agile Biofoundry, which works with industry and academia on maybe 24 or so vastly different microbes to make a variety of different end products from chemicals to fuels. They've sort of taken the approach that they want to focus their metabolic engineering efforts on a very wide mouth funnel at the front for sugar-based carbon sources, and then create specific microbes that target beachhead molecules, which are points in metabolism from which you can make a variety of different end products. So if you can efficiently funnel carbon from a very wide mouth funnel into a single product that's very close to something that we might want to use as, say, a fuel or a chemical replacement, we can make a lot of progress with a limited number of chassis strains. We also look at companies and fund companies like LanzaTech that focuses on industrial waste gases, or really any source of syngas as their carbon or energy source, which really can move this biological engineering framework beyond things that are just like just sugars to products into a variety of different kinds of intermediates that microbes can consume. Our algae platform looks at using sunlight instead of biomass as a source of energy and transforming that directly into fuels and products. These efforts are kind of more akin to traditional farming and probably encounter the most complicated regulatory landscape, in which we can talk a little bit more about later. And lastly, I did want to mention we have a bottle consortium that focuses on plastic waste and engineering novel microbes and enzymes to create biological recycling processes for plastics. So the amount of different types of carbon inputs that we can take with biology and transform them into a variety of different end products is really, really massive, and I think provides a huge opportunity space. To close, I wanted to touch just really briefly on the regulatory space. For most biomanufacturing processes in our portfolio, the bioprocessing happens in closed vessels. So at the end of a fermentation process, those spent cells are often sent to something like a boiler, an anaerobic digester, or otherwise somehow sterilized. The risk of contamination from those types of applications is really low, especially because the microbes are engineered to be super good at really specific tasks inside a bioreactor, which makes them really frail in natural environments. Some people have kind of referred to this as the cow in the jungle type of analogy, which kind of draws onto Spencer's theme. So things aren't well adapted to their environment. They're not going to outcompete the natural organisms that exist there. And some processes can actually, in fact, use their spent cells and as animal feed applications, since they're high in protein and lipids. The second case, going beyond kind of closed biocontainment, is really the case of algae, and that's very different. In that case, we work hand in hand with our colleagues at the EPA to ensure that the outdoor trials that we perform have containment in place and follow all the regulations in acts like TSCA. And the hope is to collect enough data eventually that these microbes are all safe to use and can be treated more like traditional crops. And with that, I'll close.
Great, right on time. Thanks so much, Jay. Thanks to all three of you for those opening comments. Lots of questions to ask you, including some from the chat, but I want to just start with regulation because that's been a big theme of the morning. Kent and Jay, you both spoke to some of your experiences with that, and Kent mentioned the complexity and perhaps interest in streamlining. I just want to ask, what else do you all need from the regulatory system? Given kind of the unique and complex and broad ranging amount of applications you're all talking about in this session, what would be helpful from the regulatory system? Jay, why don't I go with you?
Sure. I guess from my perspective, DOE is kind of agnostic to this. We really want to have the folks in industry that we work with be able to navigate these systems reliably and effectively so that we can achieve our goals of helping to lower things like greenhouse gas emissions. So really any clarity in the regulatory frameworks that helps our industry colleagues and partners, work with those agencies closely and effectively is what we're looking for. And I think we've often seen that, particularly in the case of algae, when we've been able to get trials approved on a variety of different genetically modified algal strains for limited sorts of deployment efforts in kind of smaller scale open ponds. And so I think that a lot of those folks have been able to effectively work with folks at the EPA to really get that to be able to happen and start collecting the data that we need to show the safety of those strains.
Okay. Thanks, Jay. Kent or Spencer, anything you want to add on regulation and what the needs are?
Sure. Just really echoing that, more clarity and guidance. As we've approached environmental applications, we've been as forthright and upfront as we feel like we can, engaging proactively with all levels of regulatory. And in our experience, we perhaps incorrectly started from a top down, for the marine work. We have a project that's, first of its kind, a biocement material with an active wild type consortia enhancement put in marine waters. So we went top down. We started to engage the EPA and telling them, what we were wanting to do, and nobody seemed to know what to do about it. They had answers for if our organisms were genetically modified, but not to that specific proposed use case. So in that experience, it seemed hazy, and it was just, okay, talk down a level, go to the state. So we engaged folks at the state in North Carolina Department of Environmental Quality. And similar response. It got down to more of a hyper-local type of decision. So, any kind of clarity and direction will help our cause when we're putting out pioneering type of applications. And it's not an easy answer because it's uncharted territory in a lot of respects. This isn't an algae installation, controlled ponds that have been implemented for decades now. So yeah, it's kind of the Wild West in a lot of respects.
That's both exciting and perhaps a little unnerving for folks. Maybe one more case in point on regulation. Jay, just really quickly, there was a question in the chat, just your mention of waste as animal feeds is just one example. Where does that fall into the regulatory system? Is it a food additive? And how are you guys approaching that?
That's a great question. I'm not super familiar with whose regulatory system that falls under. I think for non-modified organisms, those are typically a little bit easier permissions to get. I think where our stakeholders and the companies that we work with run into trouble is when they do a genetic modification that really on a kind of a that has nothing to do with, say, antibiotic resistance or something like that, but maybe is a knockout strain that's just a slight modification to a natural organism. Who regulates then whether that can be used as kind of an animal feed product? And several companies I know are trying to navigate that, because you have this really high quality source of protein and lipids that is basically just being burned because we can't or they don't know how it could be beneficially used, or if there might be side effects. So I think working closely with the agencies there would be super important.
Thanks, Jay. So we've been focusing on regulatory issues. We only have about 11 minutes and there's so many things I want to talk about. And they generally fall under the umbrella of governance, but not necessarily regulatory governance. So we mentioned the Wild West, Kent, and this idea of this kind of pioneering field. There's so many things that you could work on. And one element of governance is in terms of your own internal decision making is how do you prioritize and whose perspectives do you bring in to make the decisions on where you're going to invest, where you're going to focus, how you weight the potential benefits and risks. So, Spencer, I want to ask you, it looks like you said you're bird watching, you're looking at this kind of whole universe out there, all these communities, and how are you deciding ultimately what to focus in on, what to try? There's a question in the chat too around, just clarifying what you mean to be putting genes, looking into genetic modifications or edits in a microbe community, but how do you even start to decide what you're trying to accomplish, what's guiding your goals there?
Right. Yeah. So I think a lot of questions we get, and when we say, we have this technology that we can mobilize genes into microbes within a community context, it's like, well, what are you going to do with that, right? And I think at this point in time, before our kind of like recent work was completed, it basically wasn't, if you had a mixture of microbes and you wanted to target one of them and move some heterologous construct into that microbe's genome or delete a gene or do something like that, it was basically impossible. And so I think we're very much focused on the very basic ability to just be able to do so at this point in time, as well as improve that, so that the process that we're using is not terribly efficient. Like if you have 1 billion microbes that are all growing together, microbial cells, you might transform less than 0.01% of them. And so I think what we're very much focused around right now is we're using synthetic mixtures of microbes. So these are wild type microorganisms that are kind of model microorganisms that can be cultivated and mixed together, so that we can study how are we able to do this in a mixed context. Because that's really the difficulty, is that like if you have a microbe by itself and you transform that microbe with some DNA, you can apply some selective pressure and recover one cells of that microbe that are then transformed. In a mixed community, you don't really want to disrupt that community structure because you're trying to understand the community. So you want to transform one of those microbes with some DNA, but then you don't want to like decimate the community by using antibiotics to select your transformed bugs. So we're using kind of like sensitive sequencing methods to detect who is transformed, what percent of the community they make up, and then we're going from there. So we're trying to look at new things now of how we would increase that penetrance. And I think that's really where we're at, is very much at a development of this technology stage. As far as in the environment, kind of like what we want to focus on, and maybe I can touch a little bit on regulatory aspects, is microbes basically perform all of the biogeochemical processes around us. And Jay mentioned this, that like they have incredible capacity for metabolism, like that, when you take a breath in, 50% of that oxygen is being produced by cyanobacteria and algae that are present in oceans and lakes, and not just, trees, as you might learn kind of in like grade school. And so the fact that that's happening, anytime we're going into any natural, well, first of all, we just want to understand how these natural systems function, because, you know, that I think there's still a lot to be learned, as well as the fact that if we're going into a natural system and we're going to do something there, like, you know, put biopolymers or put, you know, spray something onto an agricultural field that's a biological that we want to, you know, do something with. I think understanding what is sort of the natural function of the microbiome in that location, you know, how disrupted is that microbiome going to get, even before doing any kind of directed editing to these natural microbiomes, you know, we want to understand just kind of like what is going on in a given place and how far from that is that going to change if we do something, and like, you know, how quickly will that revert back to normal, and how much does that impact kind of these ecosystem functions that microbes provide.
Great, thanks, Spencer. That also bridged us a little bit into, I think, the broader questions of biosecurity, whether it's for the natural environment or human health. So kind of want to call on Kent, just to answer my question of what drives your priorities, and to the extent they take you out into the natural world, how are you thinking about biosecurity?
Well, for me, what drives the priorities are the government contracts. So we have very targeted use case demonstrations with the military, essentially various flavors of hardening the ground with biology. So, you know, to date, you know, we always get the question, particularly with the marine stuff, of a concern of an ice 9 condition, right? They'll run away and cement the bottom of the ocean and destroy the planet. And you know, honestly, you know, the stock answer to that is you know, it's limited by its quorum and nutrient availability, so that you don't have this runaway condition. And that's a similar answer to you know, the terrestrial applications, where, you know, we are contained. Our application zone can only extend to where we feed it, and when we feed it, or when we stop feeding, it'll stop to grow. So we maintain some kind of control. But more longer-term longitudinal evaluations of you know, is what Spencer was speaking to about. What are the impacts to native biomes? That's something that hasn't been a point of focus in these prior works. It is coming up in current activities, where we are doing our own evaluations, and then also engaging third parties, because you know, we can't just nobody can be reliant on, you know, we say that we're good. That we're not negatively impacting these ecologies that we're executing these applications towards. So it's an ongoing developmental agenda that we're nestling in to current and future engagements with the military, and hopefully we build this data set and we can share this. And build this bank of knowledge of kind of what these current applications, what impacts, seen or unseen, are applied.
Yeah. Thanks, Kent. There's a lot of interest in the Q&A, and I think this might be there for you, Kent, or Jay, just around. And I think it relates to some of what we're talking about, what's required to actually go put microorganisms in the environment. There's a question about devitalization, and I don't know if you've dug into that question yet, maybe take a look. It seems to be that the bottom line question is, do you need to devitalize the environment in order to go put new microbes in it? Are there other ways to think about the interactions of the microbes that are already out there and the ones that you're putting out for trial? Any thoughts on that? Jay, do you have an answer to that one?
Sorry, I'm covered my mute button here. Yeah, so I think from my perspective, you know, when we're introducing genetically modified, well, not we, when the partners that we work with are introducing genetically modified organisms into, you know, open ponds in the environment, specifically with cyanobacteria or algae, you know, they're meant to stay contained kind of in those ponds. And so there's not sort of a local ecosystem to look at. What we do sort of voluntarily with these is test like local water sources that are around these types of ponds to see what the penetration of genetically modified organisms into those local water sources is. And then we also track, if those can sort of outcompete the sort of native plants that are, or native organisms that are already in those waterways. And from the limited amounts of studies that we've done sort of in that space, what we've seen is that you do see genetically modified organisms making their way into those different types of waterways, but you really don't see them outcompeting what's natively there. And so they basically make it in there, and then they sort of sit there and are for a little while, and then are sort of outcompeted by the native types of organisms that are there. And that's, I think, a pretty general path that these sorts of organisms that are highly engineered for a specific trait will follow. They're just not able to compete effectively, and they don't have large amounts of, say, transgenetic information in them that could easily shift to a different type of organism. They're really engineered specifically to have eliminated, say, side products in their metabolism. So they're kind of laser focused on one thing, and they don't do very well in the environment generally.
Thanks, Jay. I think I only have a minute or two left, so I'm going to ask a really quick targeted question just to get it out there, and then I'm going to do a lightning round of a final question for all of you. Targeted question, Spencer. Where are all these microbes coming from that you're looking at? And to the extent that you might learn something new or breakthrough about them, how do you think about access and benefit sharing back to where you're sourcing all these microbes? Is that a consideration?
Yeah, absolutely. I think, again, I'll just say that the stuff we've worked on pretty closely to develop the technology has been microbes from culture collections that were quote unquote soil derived. So these things have been widely shared, and as far as to my knowledge, I'm not sure of their original source material, but many of the things of the enrichment cultures that we've worked with, we work very closely with kind of our partners from wherever these things were derived, and generally it's been from within the United States. And there was some human microbiome samples that were from infants in St. Louis, I believe. But I guess, yeah, I'm quite concerned about if we are deriving a microbiome sample and learning something, I think particularly at IGI as well, we're very interested in wanting to kind of work closely with the people where these samples are derived from and make sure that kind of credit's given where credit's due. Yeah, but like I said, at this point in time, everything is, for the most part, stuff that's been widely available to most scientists through like culture collections and things like this. Yeah.
Spencer. Okay, well, I've been given permission for one more question. So just kind of zooming out, answer it as you will. As you think about voluntary stewardship as kind of a complement to regulation, the measures that you are taking as you think about risks, benefits that might extend beyond the bounds of what regulation usually covers, it could include ethics, equity, other social considerations, other environmental considerations. Please just share just a little more about how you think about voluntary stewardship at your setting and in your organizations and work. And if possible, just a quick example. And that'll be our last question for each of you. Let's go to Kent.
Yeah, as I spoke of before, these voluntary elements within our contracting tasks to include temporal analysis, environmental impact studies, engagement of third-party groups that come along and independently verify. We are engaging in that, and I think it's important that we continue to do so. And engage as responsibly. We're, as a company at heart, we're trying to positively impact the planet. So that MO should extend to even hard and dirty military projects that we're also engaged in.
Thanks, Kent. Let's go to Jay.
Sure. I think you can answer this from a couple of different points of view. And on the kind of more traditional biomanufacturing side, you have a lot of effort that's gone into doing things like blacklist screening for DNA that has been, sent to one of our projects to be able to work with, to make sure that, there's no DNA that we would be working with or using that could have sort of adverse effects for human health. So say, the DNA that would encode some sort of pathogen. And so we voluntarily look at that and try to create systems. In fact, the Agile Biofoundry created a tool called Bliss that looks at screening those kinds of microbes or those kinds of sequences that come in, so that we don't, end up inadvertently working with them. So there's that on the one side. On the kind of algae side and deployment side, for a lot of our projects, we have things like secondary containment around the demonstration ponds that have, newer strains in them that are outdoors, so that if there is, a spill there, it's contained and can be able to be cleaned up. We also have, encourage all of our partner projects and things to do outreach to, the surrounding community and make them aware of, the projects and what sorts of things are kind of going on with those deployed algae strains, and kind of to help kind of build a consensus around, the fact that, these things are being deployed for a specific reason, and, educate people as to what the risks and benefits might be.
Great, thanks. I see Melinda coming on. So Spencer, really quick last thought on voluntary stewardship.
Yeah, so at its core, what our research group wants to do is understand how microbial communities function in the environment, be it by using metagenomics on environmental samples or applying genetic editing in lab-based conditions. But I think really what that comes down to is, we want to understand how these things function and how they grow, but that's what I'm hoping is turned into, we want to confer that information to regulatory agencies. You can turn that into a tangible metric to say something like, well, this community or this change that's occurred after you've, done this thing in the environment is this distant from what it originally was. Like, we need some tangible understanding and metrics of how far from an original a new thing is and what that really means for us. And I'm hoping that the work we're doing can answer some of those questions. That's kind of what we'd like to see.
Great. Spencer, Kent, and Jay, thanks so much for your insights in this very fast, but I think wide-ranging conversation, and covered a lot of ground, and encourage you to look at the QA. There may be some questions in there that you can answer for the audience in writing as well. So thank you. And Melinda, we'll turn it back to you.